AlzeCure Pharma is a Stockholm-based company that is listed on First North Premier since November last year. And it's founded by a group of drug discovery professionals with extensive experience from pharma industry. And our key focus is on Alzheimer's disease, an area of huge unmet medical need, but also a multi-billion-dollar market. And we target this disease using two different platforms. The NeuroRestore platform, which is a novel symptomatic therapy, where we have the first lead candidate currently in clinical stage, and Alzstatin, which is a disease-modifying therapy. Now, Alzheimer's disease is, as you know, a disease that affects the human brain, which is the most central organ in our body and who makes us who we are. The brain consists of many different regions that communicate with each other in vast networks of interconnected nerve cells that signal and transmit information in the form of electrical impulses to each other and support important functions of the brain, such as cognitive function. Now, in Alzheimer's disease, there are these proteinaceous aggregates formed in the brain, so-called amyloid plaques. And these different aggregates are toxic to the nerve cells. So the nerve cells over time will start to shrink, shrivel, and eventually die off, which lead to the symptoms of the disease. Now, with our two projects, we target specifically these two key pathological hallmarks of the disease, namely the aggregation of amyloid in the brain, as well as the neuronal dysfunction that causes the symptom of the disease. So how about the market size for this disorder? Well, globally, more than 40 million people are affected by this disease, and the associated socioeconomic costs are extreme. So they are close to $1 trillion last year alone. And with regard to therapies available for this group of patients, there are only 4 therapies available, all of which share very limited efficacy and a lot of side effects. So it's no surprise that the potential market value in this field is astronomical. So a disease-modifying treatment is estimated to achieve at least 10 billion US in annual sales. So how about the interest from potential partners? Well, we have seen over the last years that it is a strong interest in the CNS field. This shows you some recent examples from the last 18 months in the area, showing deals made in everything from the preclinical state up to phase 2. And 3 out of the 4 here involves Alzheimer's disease. The 4th one is in Parkinson's disease, actually. But there is a continued strong interest from pharma partners in this area. And what we've seen actually is a move from, rather than having large internal R&D efforts in the area, rather in-licensing innovative opportunities from biotech industry, such as these. So how do we go about attracting partners then, and trying to build value in our company and build interest for our different projects? Well, first of all, we target specific genetic or specific pathways or biological mechanisms that have a strong genetic support that builds validity in the mechanism per se, but also increases the probability that we'll actually see an effect when we come to the patients. We're working with small molecules. These can provide cost-effective therapy because they can be given orally over a long time period at a low cost, but also provides a superior penetration rate through the blood-brain barrier than biologics do. We also work heavily with biomarkers. This is really important in defining the patient population to be treated and at the right time and with the right dose. And we work very closely here with key opinion leaders in the field to define the optimal biomarkers for our clinical trials. We're also working with a broad portfolio of CNS candidates. We have multiple candidates in our pipeline that I will show you later on, which is key both for potential partners having multiple shots on goal, but also here to be able to select the most optimal compound to move into the more lengthy and costly phase 2 trials. Another key selling point is having obviously an experienced team that with previous experience in the field. And I'm proud to say that we have a very strong management team with more than 100 years of collated experience from the CNS and pain area, and where we've taken products to the market, but also worked a lot in the in-licensing side, understanding what the partners are actually looking for. This expertise in the management team is further complemented by the board of directors, which have expertise within the development part of the value chain, but certainly also so in the marketing and strategy side, and even on the private equity side, further supporting the continuous development of the company. We also work very closely with key opinion leaders in the field. These individuals, such as Professor Bengt Wienblad, Henrik Zetterberg, John Harris, and Pete Snyder, are all world-renowned experts in their respective fields and are critical in defining both biomarkers but also the clinical trial protocols for our upcoming clinical studies. So again, we have our two platforms, small molecule platforms that we work with in this area. NeuroRestore, which is targeting patients with symptoms in later stages of disease, and Alzstatin, which is targeting patients at the early stage of the disease. Both of these programs, we do have multiple candidates within these different programs, and we have 855 as the lead candidate in the NeuroRestore platform, whereby we started the phase 1 clinical trials in December last year. We also have backup compounds to this lead candidate. This is really important because we want to make sure that we use and find the best possible candidate in moving into the lengthy and costly phase 2 trials. So we want to have more than one compound, more than one possibility to get into the patients and reach the market. This is also very critical, I would say, for a potential partner to see that we have more than one candidate. We also have candidates that are moving for opportunity indications within this area, and I'll give some more information into that a bit later. And also within the Alzstatin platform, the disease-modifying platform, we have our lead candidate 679, which is currently in preclinical development, where we are conducting safety pharmacology and toxicology testing. And here, given that these compounds are going to be given in a chronic fashion, we want to have the best, most optimal, most safe compound to move forward. So again, here we have also multiple candidates in our pipeline. So if I then turn to our first project, NeuroRestore, which is the symptomatic therapy. And as I mentioned earlier on, neurons communicate with each other through these electrical impulses that are sent along their projections. And the actual transfer of information is done at specific sites, so-called synapses. And these synapses are lost or become dysfunctional at the very early stage and are very closely correlated with the symptoms of the disease. So the synapses in Alzheimer's disease are dysfunctional and transmit at a very low frequency. NeuroRestore binds to the synapse and actually enhances the neuronal communication, neuronal function, and eventually also cognitive function. So how do NeuroRestore actually work? Well, In the brain, neurons are heavily dependent on specific growth factors. Two of the most well-known are BDNF, brain-derived neurotrophic factor, and nerve growth factor. These bind to specific sites on the cell surface and mediate important functions within the cell, such as enhancing neuronal communication, supporting neuronal function, survival, and protection. But certainly also on the system level, it supports and plays a very critical role in cognitive function. Now, in Alzheimer's disease, as well as in a number of other disorders actually, there is a dysfunction in this system that causes suboptimal signaling. And what our compounds does is actually to normalize the function of the system and thereby normalize the neuronal communication and normalize cognitive function. That's been shown, that we've shown in a number of both cellular and animal experiments. This fascinating, quite unique biological mechanism that we've studied over many years now has a quite broad effect profile, as you can see down here. And there's genetic evidence linking this system to other disorders as well, implicating that there are other indications that could be helped by therapies targeting this specific mechanism. And that's something we moved on with actually. So the primary indication for this platform is cognitive dysfunction in disorders such as Alzheimer's disease. Clearly that's a primary indication, but also in Parkinson's disease is another very interesting potential indication. And that's also the primary target for our 855 and 856 candidates. As we've shown previously in the prospectus that we had last year, we have specific compounds that are highly suitable for topical application and thereby would be highly suitable as eye drops or similar. And there are specific indications that where this system plays a critical role. So we have one candidate that's moved along in the eye and ear disorders. That's called 857. And actually yesterday we had a press release around a new preclinical project that's called TRAC-A-NAM project. And this project is again a spin-off from this platform. So we're utilizing our deep knowledge in the biological mechanism and our extensive chemical efforts in the area where we have a number of chemical compounds doing slightly different things in this biological pathway. So here we have a very interesting opportunity to move forward some of these compounds in pain syndromes such as osteoarthritis and low back pain. And this is something we've had initial discussions with pharma partners, and there's a very big interest in this area, specifically for compounds targeting this mechanism. And this is financed by the amount of money listed in the prospectus as supporting new substances. So this is something that's already financed and something that we are currently also developing. So that's not saying that we are not focusing on Alzheimer's disease, but again, trying to maximize our value for our shareholders in utilizing the assets we have within the company. And we have, again, many, many years of experience in the CNS and pain area. So this new project, the TRAC-A-NAM project, are targeting a population of patients with painful osteoarthritis that are not receiving sufficient pain relief with the current medications, which are primarily NSAIDs and opiates, which usually have a lot of unacceptable side effects. There's clinical precedence in the area whereby the development of novel anti-NGF antibodies have shown step changes in clinical efficacy in these patient groups. that really set the standard for future therapies. However, these antibodies therapies are expensive and have some adverse events related to them that we believe with a small molecule therapy we could avoid, and also at a much better cost. So here we have a clear differentiation compared to the anti-NGF antibodies. And actually we have looked at the market opportunity here, and there's very limited competition when it comes to specific small molecule inhibitors of this specific target. So here we have a very interesting new possibility and opportunity for the company. So we are moving along this project as well, although it's still in the early preclinical stages. Now back to the CD1 for NeuroRestore, the ongoing phase 1 trial for AZD855. So how do we build value? How do we attract interest from potential customers into this project? Well, we know based on experience on the in-license side, how important it is to build and to find early signals of efficacy already in the phase 1, if at all possible, to attract and trigger interest. So together with our key opinion leaders in the field, we've designed an innovative design for our phase 1 studies, which is otherwise primarily focused on safety and tolerability. That's what's required from us from a regulatory standpoint. But here, in the second part of the study, we have also incorporated signal detection study whereby healthy volunteers are being sleep deprived, and that results in a cognitive dysfunction that we can revert with our compound. So we can actually look at efficacy already in the phase 1 that we believe has the potential for us to acquire critical data that can trigger interest from pharma partners, and that's also the signal we got in the discussions we've had with them. So that's one way of trying to maximize the study design in trying to get those critical data at an early time point. The second project is targeting amyloid aggregation in the brain. And these amyloid aggregates in the brain are formed by the sequential cleavage of a protein called APP. And that's cleaved by two enzymes. This is called beta secretase, and the latter enzyme here is called gamma secretase. And this cleaves off a small peptide, which is called amyloid beta, and in the form of amyloid beta 1 to 42. It's very sticky, and over time starts to aggregate into these larger forms, oligomers, fibrils, and eventually these large-scale amyloid plaques that are so characteristic for the disease and actually what defines the disease. As I mentioned, these amyloid aggregates are toxic to the nerve cells and cause them to become dysfunctional. So this is something we want to affect. Now, our compounds, they bind to this enzyme, this gamma secretase enzymes, and cause the enzyme to cleave off a smaller peptide than the amyloid beta 42, which is not toxic and does not aggregate. And that is shown on the next slide here. So instead of forming these sticky peptides that are toxic to the neurons, we form smaller forms of peptides that are non-toxic and do not aggregate. And most importantly, we do not block this enzyme's normal function, which I think is a very critical point here from a safety standpoint. Now, coming back on the left-hand side of the slide here, we can see the actual aggregation of amyloid in the brain. So these are data that we didn't have at hand some 10 to 15 years ago. So we did a lot of clinical trials at the late stage of the disease. We didn't see much of an effect, which is not really surprising given what we today know, because here you have started a lot of secondary and tertiary events that treatment here will have no effect on. We have now seen, for example, with BioArctic's BAN2401 antibody, that going in earlier and trying to remove amyloid, then you start seeing effects. And Novartis is going even earlier, pre-symptomatically, which I think is the right way to go, given the shape of the curve. The earlier you go in here, the more likely you are to affect the actual progression of the disease. So here we want to go in at an early stage. This is the area where we're targeting with alstatin. So in early phases of the disease. And here there's enormous development of biomarkers in identifying these patients at an early stage, because obviously that's a very critical point today. Now, this amyloid hypothesis, and as I mentioned, that we're targeting the aggregation of amyloid, has a lot of genetic support. All forms of inherited forms of Alzheimer's disease is driven by mutations that cause amyloid aggregation. So there's a very strong genetic support for this. And the amyloid hypothesis was also supported then by the Biogen's phase 2 data with their BAN2401 antibody, showing that if we go in earlier and we remove amyloid, then you start seeing also an effect on cognitive function. We also want to move in at an early stage, and I would say even earlier than the antibodies. We would go in actually around here. It's more likely to get an effect, but it will also mean that we'll need therapy for a longer time period. And that's where we think a tablet or small molecular tablet will be much more cost efficient than a costly antibody type of therapy, and more suitable as well. Our compound has shown in animal models to reduce the amount of this sticky A-beta-42 peptide more than 50%. Again, has a strong genetic support. Actually, 9 out of 10 with inherited forms of the disease have a mutation in this specific enzyme. And we do not touch these enzymes' normal physiological function, which is a very important point from a safety standpoint. This is a program was originally developed at AstraZeneca, so it where we were also involved in that specific project, and we were able to actually license that project out from that period. And this is just my last slide saying that yes, we're targeting an area of huge unmet medical need where we have a lead candidate in clinical stage. We do so with 2 platforms, both targeting early and late stage of the disease, both of them with first-in-class properties based on deep science and knowledge about the genetic pathways in this specific area. And we have parallel investments in multiple candidates, so multiple shots on goal here. And we do so with a strong team with extensive experience in the field. So thank you very much for your attention.